Yeah, welcome back friends. Today we are going to continue with our topic of genetics, and today we are going to discuss regarding the chemistry of electoral material. In the previous session we discussed regarding the properties of electoral material, and in this session we are going to discuss about the chemistry or the chemical composition of electoral material. The term nucleic acid, actually electoral material, they are made up of nucleic acids. The term electoral material comes from the fact that they are found mainly in the nucleus. Nucleic acids form genetic material of all organisms. Nucleic acids are made up of subunits called nucleotides. These nucleotides join together to form a branched chain called polynucleotide. Kwa kuna nucleotide ambazo zinaungana kutengeneza polynucleotide. Hii polynucleotide ndio inakuwa nini? Nucleic acid. Each nucleotide of genetic material is composed of 3 basic components, namely the 5-carbon sugar, which is pentose sugar, nitrogenous base, and phosphoric acid, or sometimes known as phosphate group. So in terms of chemistry of electoral material, which are nucleic acid, the DNA and RNA, they are made up of 3 basic components. They are made up of nucleotides, and a nucleotide is made up of 3 basic components. 5-carbon sugar, which is pentose sugar, nitrogenous base or organic base, and phosphoric acid or a phosphate group. Let's start discussing the pentose sugar. These are monosaccharide sugar with 5 carbon in their structure. Pentose sugar. There are 2 types of nucleic acid depending on the pentose sugar they contain. Kwa hiyo aina ya nucleic acid inategemea na pentose sugar ambayo hiyo nucleic acid inakuwa nayo. Those containing ribose, yani C5H10O5, are called ribonucleic acid. Ribose, ribo. You see this? Ribose, ribonucleic acids. And those containing deoxyribose, or deoxyribose. This deoxyribose, it is because it lacks 1 oxygen. Deoxy. It lacks 1 oxygen. D means it is depleted. Ox means oxygen. So deoxyribose. Deox is because it is depleted of oxygen. So that's why the ribose sugar, it has 5 oxygen, while the deoxyribose, it has 4 oxygen. 5 oxygen, 4 oxygen. This is deoxyribose because it lacks or it is depleted of 1 oxygen. So ribose and deoxyribose are both pentose sugars. Their difference is the hydroxyl group on carbon 2 in RNA versus a single proton in DNA. So in carbon 2 of ribose, there is hydroxyl, that means oxygen and hydrogen. While in carbon 2 of deoxyribose, there is only hydrogen, no oxygen. So that's the major difference between ribose and deoxyribose. Deoxyribose lacks oxygen at carbon number 2, while ribose sugar contains oxygen at carbon number 2. In other words, ribose means the one with an oxygen at carbon 2, while deoxyribose means the one which lacks an oxygen atom on carbon atom 2 of the sugar ring. The extra O in the ribose backbone prevent formation of stable long double helical region in RNA. During formation of a nucleotide, nitrogenous base attaches on carbon atom 1. Nitrogenous base attach on carbon atom 1, while phosphate group attaches on carbon atom 5. So phosphate group attach kwenye carbon atom 5, nitrogenous base kwenye carbon atom 1. I hope you can see the structure from these sugars here below. So this is our ribose sugar and this is deoxyribose sugar. You can observe the carbon atom number 5 where the phosphate bind, carbon atom number 5 where the phosphate bind, and then the carbon atom number 1 where the base binds. You see, in the deoxyribose there is no oxygen at carbon number 2, while in the ribose there is oxygen at carbon number 2. That's the difference between ribose and deoxyribose sugar, and this leads to even the chemical stability of deoxyribonucleic acid DNA, it is more stable as compared to RNA, because this hydroxyl group, it is the reactive group, while this hydrogen, it is not the reactive group as this one. Yeah, now let's move to the nitrogenous bases after discussing regarding the pentose sugars. Nitrogenous bases, they are sometimes called as the organic bases. These are simply nitrogen-containing organic molecules which have the same chemical properties as bases. So zinakuwa ni organic molecule ambazo zina nitrogen, na kwa sababu zina nitrogen zina chemical properties ambazo zinafanana na bases. That's why they are called as organic bases au nitrogenous bases. They are building blocks of DNA and RNA. Thus, they are called the nucleic bases. Nucleic bases. There are 5 different nitrogenous bases which are divided into 2 groups. Kwa nitrogenous bases tunazivide kwenda kwenye 2 groups. Kwanza ni purines na then kuna pyrimidines. The purine bases, these are double ring compound. They are comprising of a 5-membered imidazole ring joined to pyrimidine ring at position 4 and 5. The purine bases found in nucleic acid, they are adenine, abbreviated as capital A, and guanine, abbreviated as capital G. In purines, nitrogen at position 9 of imidazole ring is the one which is linked to sugar during formation of nucleotides. So you can observe this structure of the purine, which is the double ring structure. Purine ni double ring structure ambayo ina pyrimidine ring pamoja na imidazole ring. You can observe the presence of nitrogen here at position 9. This is the nitrogen which linked to sugar molecule. Hii nitrogen ndio ambayo nitrogen nayo link kwenye sugar molecule during formation of a nucleotide. So this is the structure of adenine and this is the structure of guanine. You can observe the difference between adenine and guanine ni kwamba pale kwenye adenine pale juu kuna amine group, hapa juu kuna oxygen katika guanine. Lakini pia kwenye adenine pale pana nitrogen, wakati hapa panakuwa na nitrogen na hydrogen. Hiyo ni in terms of these purine bases. Purine bases, kumbuka, ni double ring ambazo zina 5-membered imidazole and pyrimidine. From there, the pyrimidine bases. These are single ring compound with nitrogen in position number 1, number 3, with nitrogen in position number 1 and 3 of 6-membered benzene ring. Kwa hiyo inakuwa ni 6-membered benzene ring ambayo inakuwa na nitrogen at position number 1 and position number 3. 1, 2, 3, 4, 5, 6. So position number 1 and position 3, they have nitrogen. And they are single ring structure with 6 ilikuwa inatakiwa na 6 carbon, lakini 2 carbons they are replaced by nitrogen. So we have 3 pyrimidine bases which are thiamine, cytosine and uracil. So tunachokisema ni kwamba pyrimidine bases found in nucleic acid are thiamine, which is abbreviated as capital T, cytosine capital C, and uracil capital U. In pyrimidines, nitrogen at position 1 of the benzene ring is the one which is linked to the sugar during formation of nucleotides. You can observe this nitrogen at carbon number 1, which is the one which is linked to the sugar when they are forming nucleotides. In each of these bases, there is a secondary amine, yani NH2, whose nitrogen form a bond to the anomeric carbon of the deoxyribose or ribose in DNA and RNA backbone respectively. That means you can observe this secondary amine, this nitrogen here, and this nitrogen here, a nitrogen bonded to hydrogen, and again here we have nitrogen bonded to hydrogen. All of these NH group, they are secondary amine group, and these secondary amine group, it's because the nitrogen is bonded to 2 carbon. So these secondary amine group, they are the one which bond to the sugar during formation of a nucleotide, either in DNA or RNA backbone. Each nucleic acid contains 4 different bases, 2 derived from purines and 2 from pyrimidines. Kwa hiyo nucleic acid yoyote itakuwa na base 4, 2 which are of purine category and 2 from pyrimidine category. The nitrogen in the rings give the molecule its basic nature. The 2 purines are adenine and guanine. The 2 pyrimidines are thymine and cytosine in DNA. Thymine and cytosine in DNA, with the uracil in place of thymine in RNA. That means RNA, it has uracil and cytosine and not thymine and cytosine. So thymine and cytosine is present in DNA, while RNA, it has uracil and cytosine. Thus, RNA does not contain thymine, but instead contains uracil. Thymine is chemically very similar to uracil. It is 5-methyl-uracil. Thiamine, it is 5-methyl-uracil. That is uracil with methyl group on carbon number 5. Note that the bases are commonly represented by their initial letters A, G, T, U, and C. We can move to the last component of nucleic acids, or last component of nucleotides, which is the phosphate group. This gives nucleic acids their acidic nature, their acidic character or acidic nature. That's why they are known as nucleic acids. Nucleic, it's because they are found in the nucleus. Acids, it is because of the presence of the phosphate group or phosphoric acid. So phosphate group is derived from phosphoric acid, H3PO4. Phosphate group has the same structure in all nucleotides, H3PO4. So we have phosphate there, down and oxygen, single bond, hydroxyl. It is linked to the 5 carbon of the pentose sugar by a phosphodiester bond. The phosphate group is also responsible for the strong negative charge of both nucleotides and nucleic acid, the strong negative charge. Now from there we can discuss the formation and the structure of a nucleotide. We have discussed about the components of a nucleotide. It has 3 components: the pentose sugar, the organic base, and the phosphate group. Now let's discuss about the formation and the structure of nucleotide. The combination of sugar with a base occur with elimination of water, and therefore it is condensation reaction. Combination of sugar pamoja na base, it is elimination reaction. A nucleotide is formed by further condensation reaction with phosphoric acid. 2 molecules of water are removed in the process. The similar condensation reaction occur between the sugar of one nucleotide and the phosphate group of another nucleotide to form dinucleotide. But when it involves many nucleotides, it leads to the formation of polynucleotide. The backbone of polynucleotide consists of alternating sugar and phosphate groups, with the base projecting outside away from the sugar. The sequence in which these bases occur along the length of nucleic acid chain varies from species to species and from individual to individual. Kwa sababu hizi bases ndizo genetic material zenyewe. Mpangilio wa hizi bases ndio genetic material yenyewe. Yani sugar na phosphate zinafanana, ila mpangilio wa base ndio genetic material. Unaona? It is in these bases where nucleic acid carry information controlling the organism's development. A single nucleic acid molecule may contain a total of 5 million nucleotides. The number of possible combinations is almost infinite. Now you can observe this diagram here, and this is an illustration of the formation of the nucleotide. You can see this is our organic base and this is our organic base. Now this organic base, it bonds to the carbon atom 1 of the sugar. In this case, this is the deoxyribose sugar because it has no oxygen there in carbon number 2. And then from there, after bonding with this organic base, this carbon number 5, it bonds with the phosphate group. This is our phosphate group. And you can see this phosphate group, one of the hydroxyl is bonded to another sugar there. So first, that means this phosphate group will bond to this carbon atom number 5. And then this phosphate group will bond to the carbon atom number 3 of the next sugar. You see? So this is the nucleotide backbone, which will consist of sugar, phosphate, sugar, phosphate. So the nucleotide backbone, it consists of two things. Nucleotide backbone, like this one, it consists of two things. It consists of sugar and then phosphate. Sugar and then phosphate. That's the way the nucleotide backbone is. You can also observe this phenomena here, and in this case we have a base, we have a sugar, we have the phosphate. So this is one nucleotide and this is another nucleotide. Then when they bond, that means this phosphate will bond to the carbon atom number 3 of the next sugar. So this phosphate will be attached in carbon atom number 5 and carbon atom number 3. One side it will be bonded to the carbon atom number 5, and on the other side it will be bonded to the carbon atom number 3. You see, carbon atom number 5, carbon atom number 3. Again, atom number 5, number 3. This is the way the nucleotide chain is. And the nucleotide chain, it has two ends. It has two ends. It has the third end and the fifth end. The third end is the one which ends with the free hydroxyl at carbon number 3, while the fifth end is the one which ends with the phosphate group. So it has two ends. You need to remember this, because even when we be explaining about the orientations of DNA and the RNA molecule, kuna fifth end pale, kuna third end. Kwa lazima unajua hizo vitu katika hizo molecules. But another thing which you need to know is that the phosphodiester bond, it is known as phosphodiester bond. Phosphor ni kwa sababu kuna phosphate. Diester manake bond ya kwanza hapa ni ester bond. Hii ni ester bond. Hii hapa ni ester na ile pale ni ester bond. So there are how many ester bonds? 2 ester bonds. Na ndio maana inaitwa phosphodiester, yani ester moja na ester nyingine ile pale. Phosphodiester bond, and sometimes it is known as 3 to 5 phosphodiester bond, but simply it is phosphodiester bond. Na nadhani umeelewa why it's called phosphodiester bond. So function of nucleotide, the main function of nucleotide is the formation of nucleic acid and RNA and DNA, which play vital role in protein synthesis and hereditary. Nucleotides are therefore organic molecules that serves as the monomers or subunits of nucleic acids like DNA and RNA. In other words, nucleic acid, they are polynucleotides. Also, nucleotides are used to form several important coenzymes, including ATP, cyclic AMP, coenzyme A, NADP, and FAD. These are just functions of nucleotide. In summary, if we take pentose sugar plus nitrogenous base, we get nucleoside. Then nucleoside plus phosphate, we get nucleotide. Thus, nucleotide is equal to pentose sugar, nitrogenous base plus phosphate group. You can observe the explanation in this diagram below here. We have the nucleoside made up of the pentose sugar and the nitrogenous base. This is uridine, uracil, and ribose, we get uridine. Then this is the nucleotide, uridine 5 monophosphate, UMP, uridine 5 monophosphate. So uridine simply is because of uracil. And for example, in ATP and AMP, we have adenine, but here it is used the uracil as one among the nitrogenous base. Finally, let's finalize by discussing regarding the chemical bond in nucleic acid. To discuss chemistry, to discuss in a chemical bond. In nucleic acid, there are two major categories of chemical bonds. You have phosphodiester bond and the hydrogen bonds. Phosphodiester bond occur when exactly 2 of hydroxyl groups in phosphoric acid react with the hydroxyl group on other molecule to form 2 ester bonds. So it occurs exactly when 2 hydroxyl group in phosphoric acid, they react with hydroxyl group on other molecule to form 2 ester bonds. These hold the nucleotide together in DNA and RNA. Phosphodiester bond is the linkage between the third carbon atom of one sugar molecule and the fifth carbon atom of another. Deoxyribose in DNA and the ribose in RNA. Unaona? Kwa hiyo inakuwa hii phosphodiester bond inaunganisha hapo. Let me show you here. Phosphodiester bond inaunganisha fifth carbon na third carbon. Unaona? Pale fifth carbon na third carbon. That is phosphodiester bond. So strong covalent bond formed between the phosphate group and two carbon ring carbohydrate au pentose over the two ester bond. Tatu carbon ya tano, carbon ya tatu, carbon ya tano. So ester bond, another ester bond, collectively phosphodiester bond. Another category of chemical bond in the nucleotide is the hydrogen bond. These hold together the complementary base pairs in DNA and in transfer RNA. In each case, the hydrogen bond is formed between positively hydrogen end of a polar secondary amine bond and a lone pair of electron on either a nitrogen or carbonyl oxygen. Kwa hiyo hydrogen bond ndio ile tu discuss kwenye chemistry. Ile inakuwa formed kati ya hydrogen iliyokuwa bonded to more electronegative atom pamoja na lone pair. Unaona bwana? Sasa these complementary base pair also have another important feature. A purine base, adenine or guanine, always bond to pyrimidine base, cytosine or thiamine. Yani purine moja na bondi kwenye pyrimidine moja. This means that the distance between the two strands is always the same. Kwa nini? Kwa sababu purine inakuwa ina ring mbili, wakati pyrimidine inakuwa na ring moja. Maana yake kama zingekuwa zinaungana purine peke yake zingekuwa ring zinakuwa ngapi? 4. Lakini ikiungana purine na pyrimidine maana yake ring zinakuwa 3 kwenye ile width. Kwa hiyo that's the way. This means that the distance between the two strands is always the same. 3 rings and the hydrogen bonds. So hydrogen bonds between the two purine bases, for example, would put 4 rings into the base pair and the fit would be poor. For example, in DNA, adenine form 2 hydrogen bond only with the thiamine, while guanine form 3 hydrogen bonds only with the cytosine. Guanine and cytosine base pair had 3 hydrogen bond because the exocyclic secondary amine at carbon 2 on guanine lies opposite to and can hydrogen bond with a carbonyl oxygen at carbon 2 on cytosine. The exocyclic secondary amine in guanine, exocyclic secondary amine in it is in guanine can bond with the carbonyl group katika cytosine. Unaweza kuona difference kubwa iliyoko kati ya hawa base. Hebu ona hapa bondi zinazotengenezwa mbili. Inatengenezwa bondi kati ya huyu na yule. Unaona? Inatengenezwa bondi kati ya yule na yule. Unaona? Kwa hiyo tuna yule na yule pale na yule na yule pale, bondi mbili. Lakini huyu hapa ni carbon. Unaona? Carbon hawezi akatengeneza, huyu hapa awe more electronegative kiwango cha kutengeneza hydrogen bond hapa. Lakini kwa hapa kuna nitrogen yule pale ambaye ni more electronegative. Kwa hiyo hii hapa inakuwa na bondi 3. Unaona, ina bondi 3 wakati nyingine inakuwa na bondi 2. So the reason behind is because of the nature of the structure. Kama ulivyoambiwa hapo, guanine and cytosine base pair 3 hydrogen bonds because the exocyclic amine at carbon 2 on guanine lies opposite to and can hydrogen bond with a carbonyl oxygen at carbon 2 on cytosine. Since guanine and cytosine form 3 hydrogen bonds, while adenine and thiamine form 2 hydrogen bonds, this creates a difference in strength between the two sets of Watson and Crick bases. The higher the temperature at which DNA denatures, the more guanine and cytosine base pair are present. Kwa guanine na cytosine base pair zinavyokuwa nyingi, ndivyo ambavyo hii DNA itakuwa ina denature katika temperature kubwa. Study questions: brief explain the evidence to support the idea of hereditary material being located in the nucleus. Hizo tulielezea. Explain the properties of genetic material, tulielezea. Describe the chemical structure of purine and pyrimidine, tumeelezea. Write short notes on the chemical composition of genetic material. Ndio hizo short notes. Chemical composition tasema imekuwa made up of nucleotide, na nucleotide imetengenezwa na vitu vitatu. Tutavielezea vitu vitatu. Draw the structure of adenine and cytosine. Hiyo ni swali ambalo I don't hope kama kwa mtihani unaweza kaulizwa, lakini haya maswali niliyatoa kwenye kitabu cha TAI. So let's meet in the coming session. We will be discussing regarding the types of hereditary materials katika kipindi kijacho. Thank you and later I wish you nice studies.